Hello, this is Naozumi Harada, CEO and CTO at United Immunity in Japan. The company is developing novel cancer immunotherapies using its proprietary nanoparticulate device system targeting immune cell macrophage within the tumor tissue in order to boost the efficacy of existing immunotherapies. Human cancer tissues usually contain a large number of various immune cells, including T cells, NK cells, macrophages, and dendritic cells. What you see in the right part, macrophages are the most abundant immune cells in the tumor tissue across various cancer types. More importantly, because tumor-associated macrophages, or TAMs, are the major cause of tumor immune resistance, treatment resistance. So these cells are now attracting great interest in the immuno-oncology field. In the tumor tissue, TAMs usually show immune suppressive phenotype, so-called M2 type TAMs. These M2 TAMs strongly suppress the functions of anti-tumor killer T cells, helper T cells, and NK cells by inhibiting their priming and tumor infiltration, or by inducing their exhaustion. So these effects of TAMs can lead to the limitation in the efficacy of existing immunotherapies, including immune checkpoint inhibitors, CAR and T-cell T-cell therapies, and therapeutic cancer vaccines. So my first key message is tumor-associated macrophages, or TAMs, are the next key target in the immuno-oncology field. So many companies is now intensively developing TAM-targeted drugs. They include, for example, the compounds to deplete TAMs from the tumor tissue and to inhibit the recruitment of TAMs to the tumor, as shown in the left part. However, recently, reprogramming of M2 TAMs into immune stimulatory M1 type is emerging as a novel therapeutic strategy. So for that purposes, various immune stimulants are being tested in preclinical and clinical. However, macrophages also exist in various normal tissues, as shown here, including brain, liver, lung, spleen, intestine, and other vital organs. So you have to consider the tumor-specific delivery of TAM-targeted drugs without affecting these normal tissues. But it is quite a difficult problem. Our company, United Immunity, is offering the answer. It is Pullulan nanogel delivery system. We recently discovered it is highly selective to the M2 TAMs. For example, in the mouse model as shown in the left part, we injected Pullulan nanogel intravenously to the tumor-bearing mouse and investigated the incorporation of Pullulan nanogel to the myeloid cells in the tumor tissue. And as a result, you can see our Pullulan nanogel is specifically incorporated into only to the M2 TAMs, but not to the other population of myeloid cells. And we also discovered DC sign, or CD209, a C-type lectin, is a physiological target for our Pullulan nanogel. And very importantly, DC sign is specifically expressed only in the M2 TAMs. Actually, there are accumulating evidences showing the association of DC sign in M2 TAMs and tumor prognosis across various cancer types, including gastric, bladder, renal, and lung, colon, and breast cancer. So my second key message is our device system, Pullulan nanogel, is highly specific to M2 TAMs and ideal for the drug delivery to these cells. And interestingly, our nanogel can deliver various payloads to these cells because our nanogel is consisting of Pullulan polysaccharide and also has cholesterol groups within its inside. So payload with some hydrophobicity can be loaded to the nanoparticle through the hydrophobic interaction with these cholesterol groups. And obtained complex is so-called T-ignite. So based on these mechanisms, our company is now generating our own product, UI101. So in this program, we aim to induce antigen presentation by TAMs by delivering tumor antigen peptides to these cells. This is a mode of action. Number one, if you have M2 TAM enriched but T cell excluded tumor, we will treat the tumor with our Pullulan nanogel loaded with antigen peptide. And thanks to the binding to this sign, the nanoparticle is incorporated only to the M2 TAMs. And these TAMs will initiate antigen presentation using long peptide antigen to the T cells within the tumor tissue. And this reaction is well known to induce the production of inflammatory cytokines and chemokines in the tumor, so leading to the further enhanced infiltration of the specific T cells in the tumor, into the tumor. So this tumor is now turned to the T cell enriched hot tumor, sensitive to the immunotherapies. So in order to prove the concept using preclinical models, we first generated Pullulan nanogel loaded with a peptide antigen designed for the mouse tumors. And we treated two different tumor models, mouse tumor models, with our Pullulan nanogel containing long peptide antigen or T-ignite in combination with anti-PD-1. And we used two different injection routes, intravenous or intratumoral injection, shown in the left and right part, respectively. And in both cases, we observed a good synergy between the T-ignite and anti-PD-1, and it was more remarkable in the intratumoral injection of our T-ignite with anti-PD-1, as shown in the right part. And please be noted that these tumor models are resistant to the monotherapy with either T-ignite or anti-PD-1. So we also investigate in part mode of action of the combination immunotherapy. And this is a colon tumor model, and we treated the tumor with an intravenous or intratumoral injection of T-ignite with anti-PD-1, as shown in left and right, respectively. And we detected that T cell infiltration in the tumor tissue, as shown in red. And as you can see, and in both injection routes, the combination immunotherapy enhanced the infiltration of T cells into the tumor tissue. And it is much remarkable in the intratumoral injection of T-ignite with anti-PD-1, as shown in the right part. So motivated by these preclinical POC and mode of action data, we are now developing our product. And this is a table showing the similar technologies developed by other companies. And we strongly believe our technology is much superior to others because, as I explained, our device system is quite specific to the tumor tissue or M2 TAMs. And interestingly, our device system can deliver various payloads, including peptide, proteins, oligonucleotides, and small molecule compounds. On the other hand, other technologies have some limitations in the tumor specificity or the kinds of payloads to be delivered. So using this technology, we are now developing 4 programs 4 products, 3 in immuno-oncology, and 1 is vaccines for infectious disease. And UI101, which was explained today, is at preclinical and to be developed for the combination with anti-PD-1 for the indications shown here, including head and neck, gastrointestinal, and lung cancers. And the second one, UI201, is more advanced. It is combined with TCR T-cell therapy and is already tested in clinical. I mean, our company has already accomplished the first in human phase 1 clinical trial with a combination in collaboration with our academic partners in Japan in the patients with synovial sarcoma, that is a well-known immune-resistant tumor. And we treated 3 patients with the tumor and observed 1 CR, 1 SD, and 1 PD. And the CR case showed long-lasting effects for more than 2 years. And 1 SD case was actually a mixed response, meaning the primary tumors responded to the treatment, but the new region didn't. So judged SD. And the third program is so-called Nanogel, is a kind of collaboration with Big Pharma. And actually, indeed, we are now conducting 1 program with AstraZeneca and Zyphos to generate a new nanoimmunotherapy. So this is our IP position. And we already secured our strong IP position by having these patents covering our key technology and applications. And we are so expanding the portfolio, IP portfolio by our additional applications of new patents claiming new technology and application. This is the team, management team of our company, and we have now 4 directors, including me, and I am in charge of research and development. And Dr. Watanabe is a CBO, and Mr. Diamond is IP lead. And we also have the auditor, Mr. Yaguchi. And our company has now almost 10 employees, but are now expanding our management and research and development team and aiming at 20 or more within 1 year. And in order to accelerate our research and business, we have key academic collaborators shown here, including Professor Akiyoshi in the nanotechnology at Kyoto University, as well as 3 leading scientists in the immunotherapy at Nagasaki University, UCSF, and Immu University. And our company is strongly supported by great investors, including UTech, Axil, and Mitsubishi, and others, as shown here. So coming to the end of my presentation, I greatly thank you for your attention, and we are eager to develop our nanotechnology, nanoimmunotherapy to overcome immune-resistant, advanced, hard-to-treat human cancers. So if you have any comments, questions, or any proposals of collaboration or anything else, please contact us through the website or contact us through the email shown here. Thank you very much.